Hello friends, today we are going to draw another life cycle. In this case it's going to be the life cycle of a member of the Coniferophyta, also known as the Pinophyta, which is one of our 4 extant or still living members of the group of plants known as the Gymnosperms, or naked seed plants. And just calling them naked seed plants implies that there are perhaps plants that have seeds that are wearing clothes, but they're not wearing clothes. What they are wearing, in fact, is an ovary wall around them, and those would be the flowering plants, which we should always think of as also being the fruiting plants. But that is not what we're drawing today. What we're drawing is one of these naked seed plants where there is no ovary. They're just ovules that are not completely enclosed. So the first thing we're going to draw here, I'm going to draw a line, and however you want to draw it, you might want to channel your inner Bob Ross. And we're going to draw however you want to draw it. There's going to be a series of triangles. Whatever this is meant to be. Our mature sporophyte. Squeeze that out in there. Mature sporophyte. Remembering that our sporophyte is always going to be diploid. And what is the function of a sporophyte? Its function is to make spores. That is true for all plants. All plants have sporic meiosis. So even though we might not see one part of the life cycle, we may not see the gametophyte in our vascular plants because it's hidden or tucked away inside of the sporophyte. It is there. So our sporophyte's function is to make the spores. Now, this is a seed plant, and all seed plants are heterosporous, meaning they make 2 types of spores, and only 2 types of spores in this case. We've got big spores, and we've got little spores, which are very conveniently called megaspores and microspores. And that's the function of the sporophyte, is to make these big and small spores. So in the case of our pine trees are also monoecious, which literally means one house, meaning that there's one house for both spores. So the megaspores and the microspores are produced on the same plant. In other gymnosperms, like in the cycads and in ginkgo, the large spores and the small spores are produced on separate plants, which often are referred to as male and female, which has problems. It can lead to confusion. It's not inaccurate, it's just can lead to confusion for reasons that hopefully we'll get into. Okay, so we're going to draw on here. I'm just going to draw kind of like an egg shape, and I'm going to give it some lines like this and like that to give the illusion that it has got some spirals going on. And this is going to be our Megagametophyte strobilus, also known as an ovulate cone. Or if it's a pine tree, this is the thing that we normally think of as the pine cone. So that's something we find on one part of the tree. Elsewhere on the tree, I'm going to draw kind of like smaller versions of the same kind of spirally looking thing that we drew up here, but typically found in clusters and much smaller than the Megasporangia strobilus are these clusters of Microsporangia strobili. So remember, we have one strobilus, 5, 10, 15 strobili, us, I, singular, plural, and Microsporangia strobili we can refer to as the pollen cones. So if I were to take this Megasporangia strobilus and I were to stand it up on end and section it, do a cross section through it, what we'd find is that there's a series of sporophylls, or spore producing leaves that don't look much like leaves at all, but you can tell I'm not a great artist because I'm trying to draw this very symmetrically and not doing a fantastic job. That's okay. All right, so what these things are the megasporophylls, or pointed two of them, or the modified leaves that will produce the megaspores. So I'm going to zoom in. I'm just going to indicate that that's a cross section. That's what XS means there. I'm going to draw kind of a zoomed in look at one of these megasporophylls. Okay, so here is a megasporophyll. Inside we've got the Megasporangium. We've got a covering called an integument. And we've got this gap in the integument that is referred to as a micropyle. Now, pretty much everything with the Megasporangia strobilus is referred to as mega something. It's kind of like Batman. Everything Batman owns kind of has his own brand on it, like the Batmobile and the Batcopter and the Batarang and so on and so forth. So with the Megasporangia strobilus, pretty much everything that's associated with it is mega, except for this one little thing, this gap here in the integument called the micropyle. That's the one micro thing that we're not going to find associated with the Microsporangia strobili and all that good stuff. Okay, also inside of this Megasporangium, we're going to give it a dot there. And that dot is a very important dot because that is the megasporocyte. And the megasporocyte, ending in CYTE, that tells you that it's some kind of cell. And in this particular context, in plants, when we see that CYTE, that tells us that this is going to be our last diploid cell in this generation. And when I say it's going to be the last diploid cell, that means it's going to do that special trick of going through meiosis. So let's see if I can draw pretty much the same thing again down here. Okay, we've got our megasporophyll, we've got our micropyle, we've got our megasporangium. Sometimes the megasporocyte is called the megaspore mother cell. So now not only do we Have we learned a new word for a thing we didn't know exists? We know two words for that thing we didn't know existed. The megasporocyte or the megaspore mother cell. Okay, so meiosis happens. And meiosis, being reductional division as it is, gives us haploid cells. But we have a surviving megaspore. Plus we get 3 other haploid nuclei. That don't survive. And this is because the megaspore needs all of the cytoplasm and all of the cellular apparatus to provision what is going to develop from the megaspore, which is the Megagametophyte. So we've produced one type of spore, and that's going to lead to the production of, as spores do, the production of the Megagametophyte. Okay, so what am I doing here? What am I doing? Okay, so now we've got the Megagametophyte, which is haploid. Inside the Megagametophyte are some archegonia. There are several of them in there, but I'm just going to point out one archegonium. Inside that archegonium is an egg, and that's going to be the larger of the two gametes. All right, so we're going to give that a pause to grow and develop, which is something that really happens, although instead of a few seconds, it's going to take a few months for all of that to occur. So let's focus in on our Microsporangia strobili. If we look through one of those, we'd see that they have similarly an axis covered with microsporophylls. And if we zoom in on one of those, bring it down here, what we're going to see is that microsporophyll contains the microsporangium, which is filled with microsporocytes. Now, if you are a good pattern recognizer, you will probably pick up on something that's going to happen to these microsporocytes, given that I've just pointed out that they are diploid, being part of the sporophyte. And that thing that's going to happen, just like we saw with the megasporocyte, is it's going to go through meiosis. Let me make really clear that it's the megasporocyte that's going through meiosis. But unlike with the megasporocyte, I'm trying to make it look like there's 4 times as many of these cells now as there were of the microsporocytes, because these, through meiosis, which is exciting, produce microspores, which are haploid. Okay, and these haploid microspores, like with the megaspore, are going to go through mitosis. So that microsporangium is going to end up full of these things that look like little Mickey Mouses. They look like that because we've got one circle with two smaller circles kind of on top of it. And if we want, we can put even another circle inside there. And this is our pine Microgametophyte. So we've got a primary cell which has a germ cell and a tube cell, as well as two prothalial cells. These two things that look like the ears on our mouse pig are air bladders. Because once we get into the seed plants, plants go from having a reliance on water to move the gametes to relying on air. They kind of abandon having a navy and form an air force. So these microgametophytes of only 4 cells. So mind you, microgametophyte is technically its own plant. It is its own plant. It is its own multicellular plant, even though we more commonly know of the microgametophyte as the pollen grain. Pine pollen has 4 cells. It is its own multicellular plant. It's got cells that are differentiated with different functions and purposes, which makes it truly multicellular, even though there's only 4 of them. Okay, so what's going to happen next? Let's draw one more time down here with that micropyle open. And I'm just going to make it very obvious and clear that Megagametophyte grew by mitosis. I'm going to put a couple of our pollen grains in here, even though there's going to be way more than that. I'm going to put some of our archegonia down in here. And there's usually several archegonia in there. Okay, so our pollen is going to go find that micropyle, squeeze into that micropyle, and often there's like a drop of liquid that will help stick the pollen inside there. So the pollen gets stuck, it will shrink down and pull that right into the micropyle so that the pollen tube can form. So that's what we have there. And that is going to lead to fertilization, or the start of our next sporophyte generation, right? Because we've got syngamy, fusion of 2 cells, 2 haploid cells to form one diploid cell. Now notice the way I've drawn it here. There's several pollen grains, several archegonia, and inside one of these seeds, only one archegonium is going to get fertilized by one sperm, which is going to grow into one embryo. So there's several archegonia in here, several pollen grains. but only one is going to become like the champion of the Megagametophyte to form the seed. Okay. So after fertilization, which gives us a new diploid sporophyte, at least an embryonic one. Okay, so we've got inside of here it's going to look a little bit like Forky from Toy Story. Okay. So the integuments, now that they've sealed that in there, that being the embryonic plant and the remains of the Megagametophyte, are going to form a seed coat, which is going to protect that embryo and its and the remains of the Megagametophyte. We're also going to have the remains of the Megasporangium, which is called Nucellus. The remains of the Megagametophyte become this structure called endosperm. Now, because it is remains of the Megagametophyte, and it's going to take on the role as a food source for Forky in here, which is our embryonic plant. So that when the seed first starts to develop, it germinates and we get the new embryonic sporophyte growing. It can't photosynthesize right from the jump. It needs a little bit of time to get established, so it packs a lunch with it, which is this endosperm. Okay, so let's say that our here's our embryonic sporophyte. Once it's getting out of the seed, it has an embryonic root, which is called a radicle. It has an embryonic stem, called a hypocotyl. It's got embryonic leaves, called cotyledons. And in pine trees, there are several cotyledons in there. And we have an embryonic shoot called an epicotyl. So an advantage of seeds, what makes seeds which is a great innovation for plants, is that we have a way to disperse our plants and form a new generation of plants. Not relying on spores. Now remember, spores are typically unicellular. They're totipotent cells, which means they can grow and to differentiate into any old kind of thing, any plant tissue for that particular plant that it will need, but it's just one cell. So it needs to have energy, it needs to figure out how to photosynthesize and do roots and shoots and the whole deal does a spore. But a seed, you've basically got a much better prepared for the environment type dispersal unit, because you've got an embryonic plant which already has leaves starting to form and roots and shoots and stems, plus that food supply, plus protection from the environment via that seed coat. So that embryonic plant can then grow and mature, and this is where most of its life will take place, represented by this arrow going from the embryonic sporophyte to the mature sporophyte. So the vast majority of the life cycle is happening in this arrow here. But the part that we're interested in from a testing perspective for what you need to know about the life cycle is all the rest of this. Okay? So happy pine finding to you.